Between 2020 and 2025, Withings analyzed 2.3 million sleep patterns across North America and Europe. The data revealed a 32-minute nightly reduction in total sleep time. Over 5 years, this compounds to 970 hours, 120 complete 8-hour sleep cycles eliminated from the human experience. The mechanism underlying this loss isn't fatigue. It's the progressive compromise of a waste clearance system discovered in 2012, a system that removes the same proteins found in Alzheimer's disease. This analysis reports peer-reviewed research for educational purposes and does not constitute medical advice. To understand why 32 minutes matters, we need to examine what the brain does during unconsciousness. Sleep isn't a passive shutdown. It's a meticulously orchestrated sequence that repeats 4 to 6 times per night. Each sleep cycle lasts 90 to 110 minutes and contains 4 stages: N1, N2, N3, and REM. N1 and N2 are light sleep phases. The brain produces sleep spindles and K-complexes, EEG patterns that block external stimuli. Together, they comprise 50 to 60% of total sleep. N3, deep sleep, produces delta waves. This stage drives tissue repair, growth hormone secretion, and immune optimization. N3 comprises 15 to 25% of sleep and concentrates in the first 2 cycles. REM sleep presents a paradox. Brain activity mirrors wakefulness while the body experiences complete muscle paralysis. This stage consolidates memories, processes emotions, and facilitates problem-solving. REM represents 20 to 25% of sleep and increases in later cycles. When total sleep time decreases, the body doesn't distribute the loss equally. It prioritizes. Reducing sleep from 7.5 hours to 6 hours doesn't remove a percentage of each stage. You lose entire cycles, typically the final cycles rich in REM. Matthew Walker's research documented the consequences. Subjects maintaining adequate deep sleep but losing REM demonstrated physical recovery but impaired emotional regulation and creativity. This explains why 6-hour sleep functions short-term but deteriorates cognitive markers chronically. But there's a specific function of N3 deep sleep that changed our understanding of why we sleep. A waste removal system discovered in 2012 that clears toxic proteins 10 times more efficiently during sleep than wakefulness. The glymphatic system. The human body operates a lymphatic system that removes metabolic waste. The brain, however, lacks traditional lymphatic vessels. In 2012, Mike and Nedergaard's team discovered a brain-wide clearance pathway using 2-photon imaging. They called it the glymphatic system. The G, denoting glia, support cells forming this network's infrastructure. Here's the mechanism. During wakefulness, brain cells are swollen. The interstitial space represents only 14% of brain volume. During deep sleep, brain cells contract. The interstitial space expands to 23%, a 60% increase. This expansion allows cerebrospinal fluid to flow rapidly through interstitial channels, flushing metabolic waste toward drainage pathways. Studies demonstrated glymphatic clearance operates at 90% reduced efficiency during wakefulness. The brain cleans itself 10 times less effectively when awake. The system targets toxic proteins: beta-amyloid, the protein forming Alzheimer's plaques, tau protein associated with dementia, and metabolic byproducts. During N3 sleep, these are systematically cleared. Studies confirmed that a single night of sleep deprivation significantly increases beta-amyloid concentrations. Chronic sleep restriction results in progressive accumulation. Longitudinal research documented that individuals with chronic sleep disruption demonstrate elevated Alzheimer's risk decades later. Current evidence suggests causation. In January 2025, University of Copenhagen researchers published findings revealing the precise mechanism driving glymphatic flow. They identified that norepinephrine oscillates in specific patterns during deep sleep. These oscillations drive slow vasomotion, rhythmic contraction, and relaxation of brain blood vessels. During deep sleep, brain blood vessels function as a pump, creating pressure gradients that propel cerebrospinal fluid through interstitial spaces. The study documented that GABAergic sleep medications like zolpidem reduce norepinephrine oscillations by 40%. Result: sleep duration achieved but compromised glymphatic clearance. How much sleep does an individual require to maintain optimal glymphatic function? Population averages provide a framework, but individual variation spans 90 minutes or more. Matthew Walker's research at UC Berkeley established individual sleep requirements through a two-phase protocol. Phase 1. Baseline documentation. Subjects eliminated auditory wake cues. Sleep onset occurred at genuine physiological sleepiness. Wake times occurred naturally. Environmental conditions optimized for darkness and silence. Subjects documented sleep onset, wake times, duration, and subjective quality across 7 days. The data revealed individual requirements varied by 60 to 120 minutes from population means. 73% required 7 to 9 hours, but 15% fell outside this range. Phase 2. Cycle optimization. Phase 2 applied the 90-minute cycle principle. Subjects selected target durations in cycle multiples. 6 hours equals 4 cycles, even. 5 hours equals 5 cycles, 9 hours equals 6 cycles. Calculations worked backwards from fixed wake times. Example: a subject with 6 a.m. wake time and 7.5-hour optimal duration calculated 10:30 p.m. sleep onset. Accounting for 15 to 20-minute sleep latency, bedtime occurred at 10 p.m. Phase 2 required fixed wake times daily and calculated bedtimes based on cycle multiples. Subjects achieving optimal numbers demonstrated natural wake times near alarm activation, sustained energy without crashes, and emotional stability. Subjects requiring adjustment showed alarm, snoozing, extended morning impairment, caffeine dependency, and afternoon energy crashes. Adjustments involved cycle increment changes, reducing for oversleep fatigue, increasing for persistent undersleep. Large-scale studies published in 2024 documented that 7 to 8 hours correlate with lowest cognitive decline risk. Durations below 6 hours associate with elevated dementia, cardiovascular, and metabolic risk. Durations exceeding 9 hours also correlate with negative outcomes, but these are population correlations. Individual requirements exist on a spectrum determined by genetics, chronotype, and metabolic demands. There is no universal prescription. There is individual optimization through systematic observation. Current research continues documenting long-term consequences of chronic cycle disruption and glymphatic compromise. Next analysis examines circadian photoreception, how morning light exposure alters deep sleep architecture 16 hours later. 2024 studies documented that 45 minutes of outdoor light in morning hours increased N3 deep sleep by 30% the following night. Yet 90% of individuals never implement this protocol. The mechanism will be examined in the next report.